Chapter 7, Gene Function. The learning objectives for this topic are to explain how the genotype of an organism determines its phenotype, state the central dogma of genetics, and explain the roles of DNA and RNA in polypeptide synthesis, describe 3 steps in RNA transcription, mentioning the following: DNA, RNA, polymerase, promoter, 5' to 3' direction, and terminator. Describe the genetic code in general, and identify the relationship between codons and amino acids. Describe the synthesis of polypeptides, identifying the roles of 3 types of RNA. Explain the operon model of transcriptional control in prokaryotes. Contrast the regulation of an inducible operon with that of a repressible operon, and give an example of each. Describe the use of microRNA, short interference RNA, and riboswitches in genetic control. So, in this topic, we're going to cover genotype versus phenotype, transcription, translation, and regulation of genetic expression. In talking about genotype versus phenotype, what you're used to from Biology 201 is something represented like this with the Punnett square, where you have the genes of the organism, but what the phenotype is related to whether those genes are dominant or recessive. Well, this is true of organisms that are diploid, that have 2 copies of their chromosomes. All bacteria and archaea and quite a few fungi and other organisms are haploid. They only have 1 copy of each gene. Therefore, the phenotype is regulated by whether the gene is on or whether the gene is off. This is also true of diploid organisms, because just because you have 2 copies of the gene doesn't mean it's being expressed. So genotype is all of the genes in the organism. That's basically the DNA. The phenotype is what is being expressed of the DNA. This figure also shows the central dogma. In other words, all information in the cell goes from DNA to mRNA to polypeptide. Now, when we talk about HIV, they break the central dogma of genetics because they go backwards. But we will talk about that when we talk about viruses. So, genotype are all of the genes that are in the organism, whether they're being expressed or not. Phenotype are the genes that are being expressed in the organism. So, for example, E. coli can have pili, but they don't always express the gene for pili. So the genotype is pili positive, the phenotype may be pili positive or pili negative, depending upon whether the cell needs them or not. That makes sense. So when we talked about replication, we were talking about preserving the genotype. Now let's move on to transcription, which is the first step in generating the phenotype. In transcription, we're going to be talking about some terms, and I thought I'd go over them here. Initiation. Initiation is the start of transcription, and it involves promoters, sections before the gene that tell the RNA polymerase to grab a hold here. Sigma factors are incorporated into the RNA polymerase and tell the RNA polymerase which promoters to grab a hold of and how hard. Elongation is the process of making the lengthening or elongating mRNA or rRNA or tRNA strand, and we have multiple RNA polymerases that are involved in that. Termination is when you get to the end of the gene and it's time for the RNA polymerase to let go. There are 2 types. There's self-termination and road-dependent termination. And we're going to talk about all of these in more detail. Let's look at transcription in general. Here is a gene that needs to be transcribed, and before the gene actually starts on the DNA, we have a region called the promoter. And the promoter is where RNA polymerase sits down and starts unwinding the DNA. Now, part of RNA polymerase is the sigma factor. The sigma factor is what actually binds to the promoter. Different sigma factors bind to different promoters, all of which results in different genes being transcribed more often. Here's a multicellular example, a human example, to help you get a grasp on this. So think of a farmer's market. You have all of the farmers, all of the people wanting to sell their produce, and that's kind of like all of the genes in the cell. And they have signs up saying how wonderful their fresh eggs are, how fresh and ripe their tomatoes are, how wonderful and orange their carrots are, etc., etc. Now, the RNA polymerase is kind of like me walking down the line of stalls with a shopping list in my hand. Now, if my shopping list says ripe tomatoes, I'm not going to stop at the egg stand because it's not on my list. Now, the person behind me may have eggs on their list and they're going to stop and get eggs. So it's kind of like that. It's kind of like the RNA polymerases go around looking for the promoters that match their sigma factors. So they attach, and unlike DNA polymerase, RNA polymerase can unwind the DNA by itself. It doesn't need a helicase. So it creates this bubble and starts down the gene. And this is the end of initiation of transcription. Elongation is fairly straightforward. Like DNA polymerase, RNA polymerase has to transcribe from the 5' end to the 3' end. Unlike DNA polymerase, it only does one strand of the DNA. We call that the template strand. And just like in DNA polymerization, the bases have to base pair. C goes with G, A goes with T, U goes with A, because remember we don't have thymine in RNA. We get triphosphate nucleotides coming in. We snip off 2 of the phosphate groups. That provides the energy for the polymerization. One thing to keep in mind is that previous to this, when we talked about transcription, to simplify things in other classes, we only talked about messenger RNA. RNA that was going to go on to the ribosome and be used as the information to make a protein. But there are other kinds of RNA that are functional in and of themselves, and they're transcribed too. So this same process would work for making rRNA that goes on to be a part of the ribosome. This process would also work for making tRNA, transfer RNA, that brings amino acids into the ribosome, and so on and so forth. So keep in mind that transcription includes not just mRNA. We have to figure out how to end this. When we get to the stop signal, the terminator, then we need to release the RNA polymerase and release the RNA transcript so that it can either fold into an rRNA, a tRNA, or it can go on to a ribosome to be translated into a protein. Well, RNA polymerase binds to DNA very, very tightly, so you kind of have to pry it off. There are 2 ways of doing this. One of this is called self-termination. At the termination of the gene, we have a GC-rich sequence. You have a lot of guanines, you have a lot of cytosines. Now remember that cytosine and guanine form 3 hydrogen bonds. So it takes longer for it to polymerize. Well, while it's over here slowing down, this free loop here can start hydrogen bond with itself. So the G and C's in the RNA transcript can start hydrogen bonding, and we form this hairpin. This hairpin creates pressure on the attached RNA transcript. Well, after the GC-rich region, we come to a region that has more adenines. Adenine only forms 2 hydrogen bonds with uracil, so it's got a weaker hydrogen bond than with the GC region, so it's not able to resist the pressure and it pops it right off. Isn't that clever? Now, not all genes have a self-termination region. It doesn't have a GC-rich region followed by an adenine-rich region. So we need to use what's called a row protein. This row protein kind of is circular, slides itself onto the end of the RNA transcript, and slides up, and it slides between the rRNA and the template strand and just kind of pops it right off. Up to this point, for simplicity, we've shown just one RNA polymerase going through and making a transcript. But if this is a really popular gene, we can get multiple polymerases going one right after each other, making multiple RNAs. So here are some popular genes that would have multiple transcription going on all the time. You can never have too many ribosomes. So the genes for rRNA and ribosomal proteins have promoters that match most of the sigma factors, so they're being transcribed all the time. Here's another one. You've got E. coli that has just been moved from say a glucose media to a media that only has lactose for a carbon source. Well, all of a sudden it's got to turn on genes for dealing with lactose, for getting lactose into its catabolic pathways. So all of a sudden those genes would become more popular. We're going to be talking more about that later when we talk about gene regulation. Also, if there's lots of food in the environment, the bacteria is growing and dividing, then you're going to have genes turned on and being transcribed frequently that lead to cell wall development, other structural proteins, proteins that are involved in the catabolic pathway, such as enzymes and different things. So you get the idea. There are some genes that are only needed occasionally, and so they would have promoters that didn't match sigma factors, very many sigma factors, so you might have one RNA polymerase going along it. It just depends upon the gene. So far we've been talking about transcription in prokaryotes. We've studied it most intensively there because E. coli is a wonderful lab model. So let's talk about what happens in eukaryotes. Now the lab model for eukaryotes is usually yeast, Saccharomyces. The first big difference is transcription happens in a different place in eukaryotes, inside the nucleus. Prokaryotes do not have nuclei, therefore their transcription happens in the cytoplasm. Eukaryotic genes often have sequences that are non-coding. We call them introns for intervening non-coding regions. The regions that code for polypeptides are called exons, X because they're expressed. So here is a fresh, brand new mRNA, and now we've got to modify it. One of the first things we do is we put a cap on the 5' end. It's kind of like an expiration date. Keeps the mRNA from being chewed up by RNases once it hits the cytoplasm. Also, something that acts as an expiration date is a poly-A tail is added onto the end. There isn't on the gene a transcript, a template for the poly-A tail. The RNA polymerases in eukaryotes just automatically add those in. So there are things called spliceosomes that are composed of RNA and proteins that come in and remove the introns and splice the exons together so that we get a mature mRNA that codes for one polypeptide. Now, part of the 5' cap, besides being an expiration date, also is a signal for it to be moved out of the nucleus through the nucleopores into the cytoplasm. So that's some of the differences that eukaryotes have in the processing of their mRNAs. Here are some other differences. We've already talked about place. Eukaryotes have 3 nuclear RNA polymerases and 1 polymerase that is mitochondrial. They have more transcription factors than prokaryotes have, and we've talked about the capping, the polyadenylation to the tail, and the splicing.